of fluorescent probes for diagnostics and devices. I think it's a very befitting area, and especially when we are looking at Atmanirbhar Bharat, and when we talk of 75 years of independence, I think it is very befitting that we also look at how not only in the area of diagnostics or in the area of biomedical devices, but also in the area of, let's say, bulk drug preparation, in the area of other chemicals that we generally import. How can we provide better quality chemicals within the country, manufactured in the country? I remember at one point in time, for staining of DNA, we used to use ethidium bromide, which itself is a carcinogen. But now we have developed new molecules such as CFU, which are totally harmless, and you can still use them, and they are far better in terms of fluorescence. I think that is the other thing I always feel from chemistry background. I think it's befitting that we should understand the need of the hour. We should also understand how the chemicals can be made more affordable to the Indian population. And when I say so, it means that more and more people can join the research bandwagon, especially in the university. You can get some quality research done if you are able to produce chemicals. That is one side. The other is that a lot of diagnostics do not reach the common man because of the prohibitive costs. In case we are able to develop diagnostics and devices which are cheap or affordable, I wouldn't say cheap, but they are affordable, more and more people will come in the ambit of our healthcare system. And people can actually get access to good quality healthcare, can get access to good quality diagnostics. I think that is where our institute, as well as the Central Drug Research Institute, I mean, if you look at from the point of view of CDRI, I think the entire pharma industry in this country owes its existence to CDRI and IICT and NCL3 institutes, because they were the ones who came up with processes which allowed the drugs to become more and more affordable for the Indian population. And today, when we talk of India as the hub for generics, it is only because of the fact that the scientists then, at that point in time, ensured that they would provide cost-effective processes to the company, which the company was able to scale up. And at all times, there was a hand-holding by the scientists of the institute. So I think it would be a good insight from a person known as Dr. Goel to find out how these kind of probes are developed. Not only that, but how they are also brought to a point where a technology can be made and transferred to the industry. So what are the nuances? I am sure he'll be addressing those. If not in the slide, I'll request him to please address those nuances, what the company wants, and how we address those concerns when our technology comes to the company. Hand-holding is equally important. And as a housekeeping announcement, I request all of you to please either switch off your mobiles or put them on silent. It does not disturb the system. But before I begin, I'd like to give a token of our appreciation to Dr. Goel. I request Dr. Goel to please step up, and I'd just like to give you a small memento from our institute so you remember. Not that you don't remember the institute, but at least you remember that you came here on Technology Day. I don't have a technology to offer you, but still give you a memento. And you are online as well. Okay.
My dear colleagues, friends, students, Okay. So, good afternoon, everyone. Professor Dhawan, my dear colleagues, friends, students. As you know, today is the National Technology Day. As Professor Dhawan already mentioned, that today in 1998, we did the first nuclear test at the Army range of Pokhran in the presence of Honorable Shri APJ Abdul Kalam. And then Prime Minister Atul Goel announced that day as a national technology. So I'm really privileged and thank you very much, Professor Dhawan, for giving me this opportunity on this auspicious day. For a scientist, I think nothing could be a better day for this technology. So today I'll be touching upon some of the work that we are doing at CDRI, basically in the area of drug discovery, diagnostics, and devices. So as we know that our mission is a connect to science with society, which is very, very important nowadays. Because generally we do the basic research, which is very, very important to do translational research as well. But what we found that people are just totally engaged in the area of basic research, and then they keep on doing some good publications, which is, I would say, which is very, very important. But side by side, we should also look into the what exactly we are going to give with that research to the society, which is an important aspect of what we are doing and what for we are getting the salaries and all. So I'll be just touching upon all some of these facts that we are working in the area. Yeah. Not working. Yeah. So basically, in CDRI, I'm totally involved in two major projects. Particularly, one is the one development of new leads from natural sources for the treatment of particularly in the area of bone health as a fracture healing molecules, taking lead from natural products. And another one is the diversity-oriented synthesis of new donor acceptor-based several heterocyclic molecules, which can be prepared through carbon-carbon bond reactions, and then applied to a development of technologies related to diagnostics and devices. So I'll not go into the detail what we have done related to the area of drug discovery. So I just put a kind of one slide to show the technology that we have recently given to the company. This is called the CDRI 399, which is a new medicated bone implant material to accelerate the bone fracture repair. So what we found that we found this molecule taking lead from natural product called Medicarpine, which is isolated from Rutia monosperma. And then we did some of the in vitro studies, the calcium annihilation assay, where we got very good response. And then we did some calcium labeling in vivo system and do micro CT analysis to found that this CDRI 399 is working very efficiently. So generally, what we noticed that when we have a fracture, so generally doctors when it is a broken bone, so generally people use this titanium plates, non-degradable metallic titanium and steel implants. So the problem with this is that it's a high rigidity, causes stress shielding between these bones. And another one is the inactive bone metabolism at fracture site, interferes with the radiographic imaging when we go to the MRI and all, and causes a foreign body reaction, which is really difficult to treat actually. They make a biofilms, and then no therapy can work in that site. And also, when this repair gets this bone gets repaired, then the doctor has to do again the second surgery to remove this metallic implants. So what we thought that it's a kind of next generation medicated biodegradable implant material. But we thought that if we have a kind of magnesium or strontium based, which is a biodegradable material, and we can incorporate using our own osteogenic molecules to combine to form a kind of screws and pins, we can solve this kind of issue. So the advantage of this is the excellent biocompatibility, no interference with CT, and all we can just, whatever disadvantages I have mentioned, we can just overcome through this technology. So in 2019, we transferred this technology to the company also, and we got this award. Another molecule that we are working on is the CDRI 1500 molecule. This is currently in phase 1 clinical trial at KGMO. So in this also what we found that at 1 milligram per kg dose, we got a very interesting result that the callus formation was pretty high when we treated and compared with the control. So as you can clearly see, this is a vehicle and this is at 1 milligram dose. You can see the callus formation. And this is the over-optimized model. And we are, by treatment with 1500, we are getting a callus formation to make a clear-cut bone. So this is phase 1 clinical trial is about to start very soon at KGM. And we transferred this technology to Pharmaceutical recently, and they will be taking care of the whole phase 1, phase 2, and phase 3 clinical trial at their own expense. So now I would come to the part of that fluorescent probes in diagnostics, particularly the development of indigenous RT-PCR diagnostic kits for COVID-19. So as you remember that in 2020, everybody knows the introduction. I mean, doesn't need a COVID-19 doesn't need any introduction. Just to give some of the history, the points that I have just mentioned, that COVID-19 was declared pandemic worldwide in March 2020. And on January 27th, 2020 was the first case that we got in Kerala for COVID-19. And Dr. PM Narendra Modi ji imposes this national-wide lockdown in March 25. So these are some of these different variants of COVID-19. Start from May 2020, which came in the UK first, and then after that, another one was the this is Alpha, Beta, Gamma, Delta, and this was on this basically on the second wave of COVID-19. And the third one recently we faced is the Omicron. So I'll just give touch upon a little bit about the introduction, how we came involved into this project. So when we started in 2020, I think in August or so, at that time the cost of this artificial diagnostic was very, very expensive. It was costing for a patient to around 5,000 to 6,000 rupees at that time, which was really, I mean, difficult for a person who cannot afford such type of testing. So we thought that let's do some work, because earlier we had some fluorescent chemistry based research work that was going on. So we thought to implement those fluorescent properties of those compounds to prepare an artificial diagnostic kits. So when we look into it, so we found that there is a Techman probes used in this artificial diagnostic kit, where we have a kind of at 3' end we need a quencher, and at 5' end we need a fluorophore. So that in the state form, when it is a neutral form, quenched form, this fluorophore, this fluorescence can be quenched with this quencher, and the distance between this donor and acceptor should be less than 10 nanometer. So based on this phenomenon, we try to develop some of our own Techman-like probes. And the important part is that these Techman probes are very, very expensive, and mostly are imported. So India was not making at that time. So we thought that it's a very good challenging project that we should work into it. So we tried some of these conditions. I will be showing some of the data. The mechanism is that during the polymerase chain reaction, elongation, this fluorophore breaks, and which comes as a fluorescence. So that's a readout for artificial diagnostic that we are getting. So based on this fluorophore, so there is a kind of threshold. When the fluorophore fluorescence is above the threshold, we'll get the positive signal. When it is down, that this patient is negative. So when we look into the dyes which are available commercially, generally people are using, suppose if you have to do some triplexing or multiplexing, we need 3 different dyes in different range and respective quenchers to make it as a indigenous Techman probes. So generally people are using this PAM, Hex side 3, Texas Red, PAM Red, side 5, which are ranging from this different ranges from 450 to 490, 500, 560, and then 620. So what we thought that since all these are commercial, let's try some of these different compounds where we can synthesize our own dyes. like CDRI D1, D2, D3 that we demonstrated. Similarly, the quenchers that are required, suppose for FAM, suppose people are using FAM, we need a kind of BHQ1 for that. So that is compatible for this FAM only. But rest of these dyes are compatible with only for BHQ2 or BBQ. So what we thought that since this is really challenging that all these dyes need have a different kind of quencher. So we similarly designed some of these different quenchers as well, like CDRI Q1, Q2, Q3. And when we were doing all this kind of research, we found that we got a one universal quencher. So we filed this patent. The advantage of this quencher is that we need only one quencher to cater all these four different dyes. So what we thought that let's say we can start the indigenous synthesis of this fluorescein FAM. So generally, people are using this phosphoramidite to prepare the oligonucleotide. So that is compatible. All these oligosynthesizers generally use phosphoramidite chemistry. So what we thought that, but the problem is that the disadvantage is it is not stable at room temperature, and the storage we need a -20. So everywhere it is not possible. So that stability is not there, and it is a moisture sensitive. So we thought that instead of using this one, can we use this azide chemistry, clay chemistry? So based on our experience in that clay chemistry, we thought that let's attach this fluorophore with the terminal azide, and then we can do alkyne and azide clay chemistry reaction. So the advantage of this is that it is stable at room temperature, and we can store in shed. I mean, for a year or so. And further, the other advantage is that it is damn cheap compared to what we have for the phosphoramidite. So we said it's a kind of 3 times cheaper. So, but the property is we try to do artificial experiment using those sites inside. So it was also matching nicely with this commercial dyes. Similarly, the other D3 that we were prepared. Similarly, this is a kind of phosphoramidite chemistry. So people are using the Scalfloor red 610 amidite. So what we thought that we can change it to amide, and we did all this photophysical study, and naturally we got the excitation at 502 and we can at 605. So another last one in the red region, what we found is a commercial sci-fi, and compared to that, we change some of the linkers there, and we can indigenously synthesize. All these dyes are imported and very costly, very costly. So we thought that let's develop a kind of strategy so that we can prepare some of our own dyes. So it worked very nicely. And the important point, as I already mentioned regarding the quencher, so people are using BHQ1 and BHQ2. For BHQ1, people are using for FAM, and rest BHQ2 people are using for this other dyes in the range of green, red, and orange. So based on this, traditionally 2 dyes or 2 quenchers, BHQ1 and BHQ2 are required for dry glazing. So we developed a kind of this type of unique quencher, which has a kind of broad absorption range here. So this is BHQ2, and this is the CDRI Q2 that we discovered, and it covers the whole entire region, like you can see from the green, yellow, orange, red, and also the purple. So as I mentioned that since we prepared this fluorophore, then another problem was how to attach these quenchers at solid support. So what we tried, my colleague Dr. Damodar Reddy, he tried using this. So we prepared this type of quencher with a terminal ODMT and this terminal acid to club this CPG based this solid support having this terminal amine to prepare this on solid support. So the quencher is now attached at solid support, ready for oligonucleotide synthesis. So this indigenous CPG based quencher we sent to the company to prepare the oligonucleotide of our own choice. So based on that, this is the CPG, the oligo, and where based on this, like for RT-PCR, generally people are using EG and RDRP ORS1 to determine whether the patient is COVID positive or not. So based on the sequence, primer probe sequence, we can clearly design using our CPG quencher, viral genes E and RDRP, and this azide fluorophore azide that we have left, we did the clay chemistry to prepare this type of indigenous segment probes, and then we purified this using the dual detector by HPLC, having this UV and double detector. So this, you can see, this is the FAM azide, and this is the clear cut peak with having this FAM triazole oligo CDRI CO2. So we purified this oligonucleotide, and then we tried this RT-PCR experiment in patient samples. So these patient samples are the positive RNA samples taken in the May 2021, and the experiment was done by my colleague Dr. and we got a very nice photo. So we can clearly say whether the patient is positive or negative. So in that, we transferred this technology to a biotech test private limited. And then this is the kit that we prepared, and we recently filed this patent as well. And the advantage is that it's in-house fluorescent dyes and quenchers are used for the first time in India. Actually, nobody is using right now indigenous technology. So we are the first one who are preparing the complete set of kits. So generally, you will see a lot of kits are available, indigenous kits, but all are having the imported technology. So we did from this step, starting from the quencher attached to the CPG support to prepare the oligonucleotide purification, and then we tried. So we were lucky to have this nice video. And then after that, in last year, we got the Omicron wave. So in that, it was the pressure from the headquarters that since you have already worked in this area, so why don't you work on the new mutations, the variants that we are having right now in India, floating everywhere. So just in 2 months' time, we could be able to achieve another Omicron thing, because we had the kind of capacity building already made using our past experience. So we quickly designed our primers and probes, taking 2-3 different mutations at the forward primers and the reverse primers, and we checked this primer probe sequences with the positive control, like probe 1 and probe 2, that worked very nicely. And also this Omicron-based detection kit that we developed, we validated to at the KGMU, where we got this nice results. Then you can clearly see that all these 3 patients were detected at KGMU CT values of 22. And what exactly we got from our indigenous case. So it clearly matches, and then we were very, very satisfied that it's really working in patient samples. So when we tried this, our kit to the COVID negative, we didn't get any. So that was the real sign that it's really working. So when we checked this sensitivity and specificity at KGMU, we found around 96.7 to 96.7 specificity. So we transferred this technology to the company, and company is now taking up further for taking the approval from the authorities and for commercial. So this is all about this diagnostic. So I'll just touch upon a little bit on this carbon quantum work that we are doing. So I mean, it is really simple. Suppose if you take a kind of watermelon, that's fine. You can. So what we did, we took this watermelon, take the extract of it, and some of the passive wetting agents that we tried in use, and then we did autoclave to prepare carbon quantum dots. So when we prepared this carbon quantum dots, we got some of the fluorescent material carbon quantum dots, and then we did the analysis like MBT experiment that shows that we have that nice fluorescence. Some of these compounds did the chemical analysis to check the size of particle size in that carbon quantum dot. And then we did some of this analysis using this some biometals or some other metals to see the selectivity of our carbon quantum dots. And we nicely got one net that was sensing through this watermelon carbon quantum dots. So we did some of the cell imaging studies, and indeed it was working. As you can see, this is a clear cut blue spot where we got a 10 micromolar incubation of lead. The another one that we tried is a Chico. So these are very eco-friendly materials that can be used for the developing some new product molecules. So I'm just touching upon so that those students who are not aware of this kind of, they can start doing with this. I mean, we don't need any specialized chemicals and all. Just you need a autoclave. You can react in 160 degrees centigrade and taking any kind of natural product, leaves, any plant source, any fruits, and you can just prepare carbon content and do some photophysical study. And trust me, it's really important, and then you will get a very nice compounds or carbon content which can be used in many applications. So similarly, we use the CECO and we tried some passivating agents. So the passivating agents are very important to put on this surface of this carbon quantum. So they can just interact with some of the biometals or some of the proteins or something, and then they can flourish. So based on that, we tried this life cell imaging application. So what we did, this carbon quantum dots are water soluble. So we put in a through tail vein and we monitor the life cell imaging, animal imaging of this. And what we found that this is pretty nicely staining some of the organs in that. So now we are trying to develop a kind of cancer model in mice where we can just put some of this carbon quantum down to diagnose, particularly at the tumor site, whether these are flourishing or not. So now I'll little bit touch upon the diagnostic approach for imaging lipid droplets accumulation in human cervical cancer and breast cancer tissues. So I think 4 or 5 years back, we prepared this kind of fluorescing compounds using our chemistry, ring transformation chemistry. We completed this, we purified this material in a 98 to 99% purity, and we did photophysical study. We got a nice yellow fluorescence. So when we did the cell imaging application using this fluorescent dye and taking 0.2% DMS in water, we found that in T3L1 free adipocyte at with 100 nanomolar of plant 550, we got a nice droplets. So since it was a circular in shape, we thought that it's a kind of lipid droplets. So we confirmed with the commercial dyes for lipid staining, which is a 9 red. So we tried eco-localization with this, and we got a nice result. And you can see maybe it's working. So then we thought that since it is perfectly doing the lipid droplet staining, we checked this confocal imaging in L. Leishmania donovani parasite, which is a positive agent of Cdra. So at 100 nanomolar scale, plant 552 and plant 540, other derivatives of it. These are the pictures, and then this is the Leishmania donovani chemistry goal, where one can quantify, even one can count individually that how many lipids droplets are there. So this has a several different applications in lipid metabolism and different aspects. Then we worked on this fluorescence confocal microscopic imaging in C. elegans, where we used 1 micromolar of this one, and we got we can clearly see the droplets mentioned here, and even also we can quantify in C. elegans in vivo model. So once we got this success, we thought that let's try the cervical cancer 30 patient samples, where you can see this is the plant 550 alone, and this is the cancer tissue where we get a kind of accumulation, a lipid droplet accumulation. So this co-staining was done with Nile red, and you can see this is the nucleus and then merged in it. So our observation was that in case of cancer tissue, there is an accumulation of lipid droplet. That is important to do some of the other mechanistic studies why it is so in cancer patients. So that validation was also done at KGMO, where we use some of this triple positive cancer patient tissues. So first we did this cell in the MCF7 cell lines, where we have this positive and this negative one, where we have this, you can say, the lipid droplet, nicely doing the lipid staining. Similarly, our 250 is also doing, and then this is the merge image. And then we tried this triple negative MD 231 cell lines. Similarly, we got a nice result. Lipid cross is right here, where which is detected at 590 to 648 nanometer. And for our plan 550, we use this 505 to 570 nanometer. And we got clear cut lipid droplet staining in some of these cells. So once we got that it's working in triple positive and with triple negative cell lines, we thought that let's try to give tissue sample, patient tissue sample. So we took this P1 and P2 two patients, which are one is diagnosed with this triple positive and another one is triple negative. And this is the tissue image of this triple positive. You can see this is the lipid droplet with staining and using this lipid top commercial available dye. And you can see the contrast that we are getting using our plan factor. Similarly, this is for this triple negative tissue samples, where we have this is the lipid top, our own plan 550. Then this was then we work on this cervical cancer section for some of these patient samples. So first of all, we did some of these patient samples with the term called T3 and T4 for patient 3 and patient 4 with the age group of 40 and 50, and they were diagnosed with the CSIR-2B and CSIR-3B type of cancer. So as this part was done in collaboration with KGMO, where we have this scanning of cervical cancer service stage tissue section of plan 550. This is the lipid tops, and you can see this contrast image that we got using our plan 550. So the concern is that we can use our own plan 550 because these guys are really expensive and important. So I'm trying to develop some of these kind of commercial substitutes so that in the, we can move one step ahead towards the self-reliant. So there is another one which is called the new visible light excitable vacuum staining dye. So when you check in the literature, generally people are using these three different dyes. One is the blue region, other is the green region, and this one is the red region. So this is the neutral red. This is called Cdra, and another one, this is the CMAT. So the problem is that this excitation of this dye is at 353. So it's a kind of UV range. So and the emission is 466. So we thought that since this is toxic to the cell, can we have a kind of excitation in the visible range so that it will be less toxic to the cell. So what we found that when we substituted using our different donor receptor group into it, we found that excitation is in the range of 400 to 436, and we got nice emission shift in the range of 486 to 506. So when we tried this type of doing in the S services ease, we got a nice result. So you can see this clear cut vacuum scanning using our own dye, this one 4A and 4B and 4C. So we tried some different techniques to check which one is more efficient. So this is a very interesting another project that we are doing in collaboration with FGPTA. So generally, if you see, we are trying to establish first diagnostic facility for imaging human brain metabolite plaque detection, which is there in other applications. So generally, people are using this type of PET tracer, which is a gold standard positron electron tomography tracers used to detect other patients. So this is the control image, and this is the AD patient. You can see clearly when using this strip, lack of this PIV retention in the entire gray region, you can clearly see. But when we tried the SPAP retention in the frontal and temporal cortices of this patient, you can clearly see right here. So the problem is that it is commercially, it is developed from the PETs for USA, but the problem is that the half-life, which is only a 30 minutes. So we cannot transport this from USA or some other places to India. So what we thought that can we prepare this type of paste because it's very old and anybody can prepare it. I mean it's free from this patent. So we tried that. Let's try. We can synthesize these of the molecules. So based on that, we also tried some of the different derivatives. So we succeeded some of them, and we have already prepared this one. And first patient sample we have tried using, and then you can see this is the normal patient, and there is like similar to this one where we didn't find. So, but the problem is that that COVID-19 came at that time, so we could not able to move further. Very recently, we came to know that machine is still working at SDPDI. So now we are about to start this work, and I'm pretty hopeful that it will definitely work and will be an asset for our institute as well as India. So this is so far about the diagnostic part that I have discussed. Now I'll just touch upon a little bit chemistry related work which we are doing using this diversity oriented synthesis of noble donor acceptor based organic fluorescent molecules which can be prepared through carbon-carbon bond formation reaction to prepare some of the organic electron. So this is just a introductory slide to the students that you know that carbon-carbon bond formation reaction are very, very important and crucial and integral part of all the organic synthesis research. Generally, people use two types, non-metal catalyzed and another one is a normal reaction. People are using this organometal catalyzed reaction because these are the non-metal and azole reactions, and in this category, CSIR-CDRI and all organometal catalyzed. So it was interesting to read one of the articles from the Nicolau in Nature in 1994. We got, when we read this interesting molecule, interesting paper, we found that they have prepared this ring C of Taxol using very simple this electron chemistry, where they use diene, react with dienophile in boronic acid, will get this type of intermediate, and then endodenes at a reaction, we can prepare this type of molecule, and based on rearrangement, we can prepare this ring C of Taxol. So generally, when you look into this simple chemistry, diene reacts with dienophile to prepare this type of hydride, and on elimination of carbon dioxide, we'll get two different type of isomers, which is really difficult to isolate. Similarly, if you use this one and silicon hydride to react in thermal conditions, we get endo-exo products, which are really difficult to isolate. So in this case, the reactivity and selectivity was the issue, and generally reactions occur at high temperature, and undesired products are formed, which are difficult to isolate. So we thought that can we move from complexity to simplicity? Can we devise a kind of methodology where we have a very simple, easy process, and there will not be any issue for selectivity, and that reaction can work at the room temperature. So this was our objective. So we thought that let's apply a concept-based approach where we can try and apply. If it works, we can, based on the results, we can modify some of the reaction conditions. And once we got the success, we can explore and we can make diversity into it. So simple, if you see this topology of this 2H one, this is a very simple stupid molecule, but it generates wonders actually. So when we see this molecule, so that C2, C4, and C6 positions are electrophilic in nature and susceptible to nucleophilic attack. So when we thought that, so these three positions can, if we put a kind of electron withdrawing group at position 3, we can activate this position because this is the conjugation. And similarly, to reduce the electrophilicity at this 4th position, if we put a kind of electron donating group, we can really activate this position 6, which is susceptible to nucleophilic attack. So based on that, we designed some of this donor acceptor based 2H param to ones, and we tried conversion of this to convert to the benzene, simple benzene thing. So we want to identify this, what are the base conditions and temperatures required to do carbon-carbon conversion, where we can generate this type of diversity with different substituents at this position. So we thought to explore this benzene by arrays, teraryls, tetraryls, and quaternaryls. So the very first reaction what we tried, we prepared this type of donor acceptor based with parent bond to react with this melanonitrile in simple PLS DNA, we could able to achieve this benzene in an efficient manner. So based on this, we tried some different active between compounds to generate a diversity, and here you can see these two aminopyridines can be prepared using the cyanamide. And some of this type of active between where we can introduce to methyl group, which are sometimes very, very important, and this isoprenyl unit and then second. So based on that, we generated kind of different. And interestingly, but when we tried this trimethylsilane type of this acetyl trimethylsilane, we could able to form a carbon-carbon bond. So based on this, we tried different type of reactions. I will not go into the detail of this. So these type, whatever we have proposed, we could able to achieve all this in a nice, simple, doing RT and using these simple reaction conditions like KY, BMF, NaH, or simple base at room temperature. So as you can see clearly, we can generate a diversity of, and you can see, suppose it is really difficult to prepare 1, 2, 3, 4 tetraaryls. People can use some organometal catalyzed reaction, but it is really challenging. But when you use this type of lactones and do some deoxybenzoin reaction, we can prepare this in a very efficient manner, very quickly. And when there is a kind of interest, we can do some kind of atropisomerism that we got, and then we can separate it out. So since we thought that since we have a very good methodology where we can just modulate all this type of reactions, or we can prepare different heterocyclic or aromatic compounds, so we can apply this type of conditions and to develop a kind of natural production process. We can do some of this. We can prepare some of these efficient ligands for catalysis. Some biologically active molecules can be prepared using this methodology. Diversity can be generated. And these type of molecules can be used in organic electronic devices, fluorescent probes in life cell making applications, and chemosensor. So we thought that since we have methodology, we try to develop different reaction conditions and different behaviors to prepare this type of different molecules. So as you know that fluorescent compounds are very important in the case of chemical biology, materials science, and chemosensor. So generally people are using FITC for doing the labeling of proteins and all, or similarly this calcium and tetracyclines are used to stain this bone formation at the fracture site or some other cell imaging application. We can use it. And another is a very important part, like people are now using the organic light emitting materials. So that can also be prepared like organic TVs and then car radios and mobiles. Generally, people are having this blue, red, and green LEDs. So I'll just touch upon a little bit upon this organic electronic work that we have initiated, I think, maybe 6-7 years back. So just for the students, what is OLED? It is an organic light emitting diode. So it is an organic light emitting diode. It's a thin film diode in which the emissive layer, which is this one layer, this consists of an organic compound. So this is the organic layer where we have an anode and a cathode, metal cathode. And this is a glass substrate. So through the recombination of hole, which is comes from this side, IQ, and the electron from this cathode, the energy is generated and comes in the form of a light. So when we saw the literature, we found that generally 3 different colors for full color flat panel monitors, 3 different colors like RGB colors are required to prepare a complete full panel monitor. So when we check the literature, we found that green and red with high external efficiencies are available in the literature. But the substances which are suitable for long-term use as a blue light emitters are still far from the standard of thermal stability and purity. So generally they are 8 to 10%. So why not blue, pure blue? What is green emission defect? So I'll just discuss. So during the device operation, maybe it's not working here. So during the device operation, these polyfluorines are generally used for the blue light emitting materials. So when they do use these polyfluorines and to prepare a kind of devices in it, and when they tried this devices using this polyfluorine, there was a conversion of this fluorine to fluorinone. So these fluorinones show emission in the yellow region. Generally, the purity was disturbed at that time. So nobody was understanding why there is a problem, that why we are not getting using these polyfluorines are showing very good and reaction, but when we do the device operation, it's converting into this. Some of these fluorine moieties can be converted into fluorinol, which shows yellow color emission. So what we thought that this fluorine shows emission in the blue region, and this fluorinol shows in the green region. Then when we combine this fluorine and fluorinol, we'll get a mixture of blue and green color. It will not be a pure blue color. So what we thought that can we put a kind of novel approach where we can use different analysis and modulate some of these chromophores. So even if some of these fluorine moieties can be converted into the fluorinol, that fluorinol should also work in the blue region. So this was our idea. So we tried this fluorine, we put some different donor receptor and chromophoric functionalities into it, and we got a very interesting result. So we started with Cdra to react with acetophenone and tried the MSO to prepare this lactone, and we replaced this SME with the amine, and then we tried to react with this indinone to prepare this type of fluorinol with the donor receptor functionality. So similarly, when we use two indinone, we can prepare this type of molecule. So when we did photophysical studies of these two series, one is this one, another is this one, you can see the compound 6 shows blue emission. When we converted it fluorinol, we got a kind of yellowish or green fluorescent. Similarly, when we tried this fluorine, we got a nice blue color. But when we tried converted this fluorine to fluorinol, some of this fluorinol also shows emission. So that we really want actually. So if we prepare some of the device using this fluorine, even if some of this fluorinol will be converted, fluorine will be converted to fluorinol, we will get a pure blue material because it's also into blue material. So this was our novel concept, and we patented this in US. So clearly you can see this fluorine shows emission in this 460 region, this fluorinol in the 520 region, similarly 481, this one, and when we tried this fluorinol, we got the 470. So if you mix this to 481 and 470, we get a blue color. When we mix this one, we got a kind of greenish blue material. So we did all this experiment. We did device preparation in IIT Kanpur with Dr. R.S. Anand. He's an electrical engineer, so who prepared this device for us, and you can clearly see this shows a kind of greenish blue color. But when we tried using this fluorine, we got a nice blue with a high intensity. So one way was to kind of use our donor acceptor based chemistry. Another was that can we this is the fluorine. If you kind of block this so that we can remove this from fluorine to fluorine, that cannot be converted. So we thought that let's put a kind of enzyme ring over here and try to do some of this donor acceptor functionality putting on it. And similarly, when we tried this donor acceptor functionality in chromophoric group using our lactone to convert this ketone to get this fluorine type of compound. So when we tried different donor acceptors, we found that when using SME and COE group at this position, we got a blue color. But when we changed this SME to a donor amine group, secondary amine, and then electron withdrawing using this AN, we got nice yellow color. So there was a kind of red shift. We did some electrochemical property investigation of those molecules, and in this case, we got a kind of large band gap. But in the case of this one, we had no more sticks, and then we got a less homo level, and then this shows in the yellow. So based on this, using this fluorine 3, we did prepare a device. Using this one, you can see clear cut of fluorescence in yellow color. Where we use this IPO, we got, and then this is the NPP, which is a kind of pole transporting layer. This emissive layer, and then this is the electron transporting layer, this one, and then the cathode. So during device operation, this is a real device picture of material using our emissive fluorescent molecule. This picture is there. So at that time, Dr. T.K. Chakraborty was the director in CDRI. So we thought to prepare a kind of fluorescent nameplate using our own molecule at IIT Kanpur. So this is a nameplate using our own compound. And the important part is that it's a low voltage driven device. So using this 9-volt battery, you can operate this. So it's a basically energy saving, energy conserving devices. We can prepare using our own, like OLEDs that generally people are using. It's a very less energy consuming devices, displays. So similarly, we prepared different type of molecules, tetracyclic molecules, and did some photophysical studies. Another one was this type of benzoyl derivative, and we tried different chromophores. We did some of the conjugations, doing some benzene over here, doing some biconjugation, and some of the halogen derivatives. So different type of devices were prepared, and we could able to prepare using this type of molecule. We could able to prepare fluorescent display. This is a kind of I think 6 to 7 inches long. So at that time there was a seminar going on at IIT Kanpur. So they prepared using our own molecules. They prepared this seminar on micro solar energy at IIT Kanpur. So this is the another different type of molecules that we prepared. We did some of the photophysical, we prepared the devices. You can see this clear multicolor devices that we prepared, and we did all these studies, and the stability was really pretty good in all the cases. So far, as I have already mentioned regarding this probes in the diagnostic and devices application. So using our simple chemistry, one can do. You don't need specialized chemical reagents. The important part is that even an economically way you can prepare diversity or impact synthesis, and you can apply those simple compounds to real application. So some of this one we got a very nice by using this solar effector. We could prepare red, yellow, green, and blue type of different material just by changing those effectors. So this recently we have prepared a white light devices using this simple electrode over there. And another one is we have developed this hydrogen sensor. You can see this shows the green color, and when we add this hydrogen, it converts into the blue color. So this is a light hydrogen vapor. You can see a nice fluorescence. This we have already touched upon. So I think this is all about. So there is a quote for particularly for the students. Don't be a parrot anymore. Be an eagle. A parrot talks way too much, but can't fly high. But an eagle is silent and has the willpower to touch the sky. So my humble submission is that don't be a parrot, don't talk too much. And I think it's very important to do sincere research work and keep on doing. If you devote your work and you will sincerely do, definitely you will achieve the goals. So thank you all for your time. I'm really happy to take the questions. Yeah, please.
So, the RT PCR like normal process is fluorescent
Right.
and a quench.
Yeah, right.
The mechanism is to frame.
Yeah.
So that is like distant.
Right.
Distance
Less than 10 nanometer.
Yeah. So now when you are like putting your molecule, so how it is ensured that
So we did some of the computational studies where we can just check whether it is coming into the range or not. So once it is there, and then we are getting the nice results already. Suppose if it is mismatching, so we will not get that result, artificial result. So based on that computational studies and all, then we find that it's really working, and then
and I think the original process is the
phosphoramidite chemistry. People are using everywhere. If you see this, it's a phosphoramidite people are using.
Yeah. Now you are doing the
click chemistry. This is the first time for COVID-19 detection we are using.
Is there any concern like you are using like that copper agate, the biological system? Like,
No. No, it's a nucleotide now. It's a nucleotide, and we are purifying through HPLC. We are not using, and it's a diagnostic. It's not we are consuming in human. So it's a diagnostic. They should not be problem.
Yeah. The other. I have one.
Yeah, please.
Regarding you that your Alzheimer's disease diagnosis, and you are wanting to show that that molecule with one having the carbon 11.
Yeah.
Carbon 11
That's
is
right.
the N-methyl.
Yeah.
So your molecule is also N-methyl.
No. That N-methylation with carbon. We are doing at CDRI. Cyclotron that they have.
Yeah, that's why you have to generate the carbon.
Yeah, carbon there, and then it could directly go to the patient directly from there.
Yeah.
HPLC purification and then directly go to because they have like a 60.
Yes, only in 30 minutes. So all the reaction process has to be done within the. So your probe is that that methylation can be done within the 15 minutes.
Yes, 25 power 10 minutes.
Yeah.
Very. No. I mean, once it is prepared, directly you can just go to the.
Yeah. So what methylating agent you are using in this case?
I'm not using. I mean, my colleague, actually my first student.
You have done it with the.
They give the free amine and then they attach this amine. They do the carbon labeling.
Carbon labeling has to be with the substrate.
Substrate.
Carbon 11.
Yeah.
Make that amine with the.
So they have that reagent.
Yeah.
Generally, people are using, I think, C-labeled methyl halide.
One is that one is used very commonly for carbon dioxide or carbon 11 carbon dioxide kind of thing.
For this is, I think,
or is the
labeled,
formaldehyde maybe
something.
because formaldehyde can do the your reductive amination easily with that or methylation. That is a very rather. This is a really excellent. Any questions from others? I must from myself or my personal and from the institute, I must say a great lecture to hear that required a different part of chemistry, natural chemistry, organic chemistry, and that widely used in different directions. That is the diagnostic part, device part. And this is not this is a very good interesting. It's simple chemistry, but having a wide application. So I hope everybody enjoyed this excellent lecture, and we must thank and give a big clap to Dr. Goel with his excellent talk.